For over 40 years, scientists have been locked in a relentless battle with one of humanity's most cunning viral foes, the human immunodeficiency virus, or HIV. The ultimate goal has always been an effective vaccine, but this pursuit is like trying to hit a target that's constantly changing its shape, its color, and its location, all at once. The primary reason for this immense challenge is the virus' extraordinary ability to rapidly change its genetic makeup. Today on Fun Facts Online, we're diving deep into the intricate world of HIV's genetic diversity to understand why this virus is such a master of disguise and what cutting-edge science is doing to finally pin it down. At the heart of the difficulty is HIV's remarkable genetic variability. Unlike many viruses that are relatively stable, HIV is a shapeshifter, constantly generating new versions of itself within a single infected individual. This isn't a slow generational evolution. It happens at a blistering pace, and it's driven by several key factors. First, let's talk about HIV's replication engine. HIV is a retrovirus, which means its genetic code is stored as RNA. To multiply, it must first convert its RNA into DNA inside a host cell, a process carried out by an enzyme called reverse transcriptase. The critical issue here is that this enzyme is notoriously sloppy. It completely lacks the proofreading mechanisms found in our own cells' DNA replication machinery. As a result, it makes frequent errors, introducing mutations into the viral genome at an astonishingly high rate. It's estimated that HIV's reverse transcriptase introduces between 1 and 10 mutations per genome in every single replication cycle. In just one day, a person with an active infection can produce billions of new virus particles, each potentially carrying a unique set of genetic variations. To put this into perspective, the amount of genetic change HIV generates in a single person in one day is comparable to the amount of genetic change the influenza virus generates across the entire global human population in a whole year. That's an evolutionary sprint happening inside one body. Adding another layer of complexity is the process of recombination. When a single cell is co-infected with two or more different strains of HIV, which happens frequently, their genetic material can get mixed up and shuffled during the replication process. Scientists have called this viral sex. It results in the creation of entirely new hybrid viruses, known as recombinant forms, which can have a combination of genetic traits from their parent viruses. This process further accelerates the diversification of the virus, creating an even wider array of viral variants for the immune system to contend with. But our bodies don't just stand by passively. We have our own antiviral defenses. Our cells possess a family of proteins called ApoBEC3, with ApoBEC3G being a particularly potent weapon against HIV. ApoBEC3G can get packaged into new virus particles. Then, in the next cell the virus tries to infect, it unleashes a barrage of mutations into the viral DNA during reverse transcription, a process called hypermutation. This can be so damaging that it completely inactivates the virus. However, in this evolutionary arms race, HIV has evolved a countermeasure, a protein called VIF. VIF's job is to target our ApoBEC3G proteins for destruction, effectively neutralizing this line of defense. The battle isn't always a clear win for the virus, though. Sometimes, VIF only partially blocks ApoBEC3G. This leads to a lower level of mutation, which isn't enough to kill the virus, but is enough to change it. Paradoxically, our own defense mechanism can end up helping the virus evolve, creating new viable variants that might be better at evading the immune system or resisting drugs. The consequence of all this high-speed mutation and recombination is that within a single infected individual, HIV exists not as a single entity, but as a quasi-species, a complex and dynamic swarm of genetically related but distinct viral variants. This swarm constantly competes and evolves, with the variants best able to hide from the immune system and antiretroviral drugs being the ones that thrive and propagate. This incredible diversity within one person is just a microcosm of what's happening on a global scale. The genetic variation of HIV across the world is simply staggering. First, there are two main types of the virus, HIV-1 and HIV-2. HIV-1 is far more common and pathogenic, responsible for the vast majority of infections and the global pandemic. HIV-2 is less easily transmitted and is mostly confined to West Africa. Because of its global impact, our focus is on HIV-1. But even HIV-1 isn't a single thing. It's divided into four distinct groups: M, N, O, and P. Group M, which stands for main, is the Goliath, responsible for over 90% of all global infections and the cause of the pandemic. Groups N, O, and P are much rarer and geographically restricted, primarily to West Central Africa. Even within the dominant Group M, there's a bewildering amount of diversity. It's split into at least 9 different subtypes or clades, designated by letters A, B, C, D, F, G, H, J, and K. The genetic difference between these subtypes can be as high as 35% in the amino acid sequences of their envelope proteins, the proteins on the virus' outer surface. For a vaccine, that's like trying to design one key to open 9 completely different locks. The distribution of these subtypes is also geographically patterned. Subtype C is the most common subtype globally, accounting for nearly half of all infections, and is particularly prevalent in sub-Saharan Africa and India. Subtype B is the dominant strain in North America, Western Europe, and Australia. Other subtypes are prevalent in other regions. And, to make it even more complicated, in regions where different subtypes co-circulate in a population, they can recombine through that viral sex process we mentioned earlier. This creates what are known as circulating recombinant forms, or CRFs. There are now over 120 identified CRFs, each with its own unique genetic makeup stitched together from different parent subtypes. This immense, sprawling global diversity means a vaccine designed to be effective against subtype B might be completely useless against subtype C, or one of the countless CRFs. This is the central roadblock to developing a single, universal HIV vaccine. So how exactly does all this variation allow the virus to so effectively outsmart our powerful immune system? A vaccine's primary job is to train our immune system, specifically our B cells and T cells, to recognize a specific part of a pathogen and then launch an attack. But HIV keeps changing the very parts our immune system is trying to target. The main target for vaccine-induced antibodies is a protein on the virus surface called the envelope glycoprotein, or ENV. This complex protein is what HIV uses to latch onto and infect our immune cells, specifically CD4 T cells. But the parts of the ENV protein that are most exposed and easiest for antibodies to see, the so-called variable regions, are also the parts that mutate the fastest. HIV constantly changes its face, creating escape mutants that the body's existing antibodies no longer recognize. It's a classic bait and switch. Early vaccine candidates that used single, simple ENV proteins failed precisely for this reason. They taught the immune system to recognize one specific face, but the virus quickly changed its mask. To make itself even harder to see, HIV has another defense: a glycan shield. The virus cloaks its surface in a dense layer of sugar molecules, called glycans. Since these sugar molecules are borrowed from the host cell during viral assembly, our immune system generally recognizes them as self and doesn't attack. This sugary coat effectively camouflages the more stable, conserved parts of the ENV protein that would otherwise be excellent targets for antibodies. This glycan shield is so dense, it accounts for about half the mass of the entire ENV protein, acting like a cloak of invisibility. The structure and density of this shield can also vary between different HIV strains, adding yet another layer of evasion. But the immune system has more than just antibodies. It has T cells. Cytotoxic T lymphocytes, or CTLs, are another crucial weapon. Their job is to find and kill cells that have already been infected with a virus. They do this by recognizing small pieces of the virus, called peptides, which are presented on the surface of the infected cell by special molecules. But just as it mutates to escape antibodies, HIV also rapidly mutates the very peptides that T cells are trained to spot. An infected cell can thus avoid being flagged for destruction, allowing the virus to persist and continue producing new copies. The constant evolution of viral variants that escape T-cell responses is a clear footprint of the immense pressure our immune system puts on the virus, and the virus' ability to keep slipping away. Finally, HIV employs an even more insidious strategy: latency. The virus can integrate its genetic material directly into the DNA of our own long-lived immune cells, particularly memory T cells. There, it can remain completely dormant for months or even years, totally invisible to both the immune system and to antiretroviral drugs. These latently infected cells form a hidden, silent reservoir. They can be reactivated at any time, leading to a rebound in viral replication if treatment is ever stopped. A truly effective vaccine would ideally need to generate an immune response so rapid and potent that it clears the virus before this latent reservoir can ever be established. Despite these formidable challenges, the fight is far from over. A deeper understanding of the virus and its weaknesses has led to a new generation of incredibly innovative strategies aimed at hitting this moving target. One of the most promising avenues of research is the study of broadly neutralizing antibodies, or bNAbs. These are extremely rare, superpowered antibodies that are produced naturally by a small percentage of HIV-infected individuals, often referred to as elite neutralizers, after several years of infection. Unlike typical antibodies that only neutralize a narrow range of HIV strains, bNAbs can neutralize a wide variety of HIV-1 subtypes from all over the world. Scientists have isolated hundreds of these bNAbs, and they have revealed key sites of vulnerability on the HIV-ENV protein, regions that are relatively conserved across different strains because they are essential for the virus survival. There are 5 main weak spots on the ENV protein that these bNAbs target. They include the CD4 binding site, the critical spot the virus uses to attach to cells, specific loops at the very tip of the protein, a region on another loop that is tied to a specific glycan, the junction between 2 protein subunits, and a highly conserved region of the protein stem that is exposed just before the virus fuses with a host cell. The ultimate goal, the holy grail of HIV vaccine research, is to design a vaccine that can teach anyone's immune system how to make these powerful bNAbs on command. This is incredibly difficult because the precursor B cells in our bodies that have the potential to produce bNAbs are rare and often don't recognize the HIV-ENV protein very well at first. To overcome this, scientists have developed a brilliant strategy called germline targeting. It's a multi-step immunization process designed to mimic and accelerate the years-long process seen in elite neutralizers. The first vaccine shot, the prime, uses a specially engineered immunogen designed to specifically bind to and activate only those rare precursor B cells. This is followed by a series of booster shots, each with an immunogen that is progressively more similar to the native HIV-ENV protein. This step-by-step process is designed to carefully guide the maturation of these B cells, encouraging them to develop the necessary mutations to produce potent and broadly neutralizing antibodies. Early clinical trials of this approach have shown it can successfully activate the right starting cells in humans, a major proof of concept and a huge step forward. To tackle the global diversity problem directly, Researchers are also developing mosaic and multivalent vaccines. A multivalent vaccine is a more straightforward idea. Simply combine antigens from several different HIV clades into a single vaccine, hoping to induce an immune response that provides broader protection. Mosaic vaccines are more high-tech. They use powerful computational algorithms to analyze thousands of viral sequences from around the world. From this data, they assemble artificial mosaic antigens that are a patchwork of the most common genetic fragments found in circulating HIV strains. The goal is to maximize the coverage of potential T-cell targets, thereby inducing an immune response that is more likely to recognize whatever strain a person might encounter. While a recent large-scale trial of a mosaic vaccine, the mosaic trial, was unfortunately discontinued due to lack of efficacy, these approaches are still being refined and explored, often in combination with other strategies. And of course, we cannot talk about modern vaccines without mentioning the new technologies that have been absolute game changers in recent years. The mRNA vaccine platform, famous for its rapid development and deployment against COVID-19, has emerged as a powerful and flexible tool for HIV research. These vaccines deliver a piece of genetic code that instructs our own cells to produce a specific, carefully designed HIV antigen. This allows for the incredibly rapid design, production, and testing of new and complex vaccine candidates, such as those needed for germline targeting. Several mRNA-based HIV vaccines are currently in early-stage clinical trials, and some have already shown the ability to induce neutralizing antibodies. Another exciting approach is nanoparticle vaccines. These platforms offer a way to present antigens to the immune system in a more effective way. Scientists can engineer these tiny particles to display multiple copies of an HIV antigen in a highly organized and repetitive manner. This structure closely mimics the surface of a real virus and can elicit a much stronger and more durable immune response than simply injecting free-floating proteins. Nanoparticle vaccines have already shown they can successfully stimulate the production of B cells that can lead to bNAbs in animal models, and a self-assembling nanoparticle HIV vaccine showed promise in a Phase 1 clinical trial by inducing a robust T-cell response. The path to an effective HIV vaccine is undoubtedly long and fraught with challenges. The recent discontinuation of some large-scale clinical trials has been a sober reminder of just how difficult this task is. But it's critical to understand that these trials are not failures. They are essential learning experiences. Each one provides invaluable data that informs the next generation of vaccine design. The central obstacle remains HIV's astonishing genetic diversity, its uncanny ability to be a perpetually moving target. But the future of HIV vaccine research lies in a multi-pronged approach that combines our ever-growing understanding of the virus weaknesses with these cutting-edge technologies. The focus is shifting towards more sophisticated strategies that aim to induce those rare but powerful broadly neutralizing antibodies through guided, step-by-step immunization, to elicit robust T-cell responses that can recognize a broad range of viral variants, and to harness the incredible speed and flexibility of mRNA and nanoparticle platforms. The quest for an HIV vaccine is a marathon, not a sprint. It is a testament to the resilience, collaboration, and ingenuity of the global scientific community. While the target continues to move, our aim is getting better. Our tools are getting sharper, and our understanding is getting deeper. Through a combination of basic science, innovative vaccine design, and international collaboration, the world is closer than it has ever been to developing a vaccine that could finally bring an end to the HIV pandemic. Thank you for listening to Fun Facts Online.